Thank you, ma'am. Thank you, sir. Good afternoon, everyone. We welcome you for the first technical session of the day one of AICTE-ISTE 6 days online refresher program, Leveraging Academic Research Lesson Learned from COVID-19. I would like to take this opportunity to express a hearty welcome on our institute's behalf to our guest speaker for this session, respected Professor Dr. Vandana Patravale, Madam, well-known academician, passionate researcher, innovator, and recipient of various prestigious awards in pharmaceutical sciences. It gives me immense pride and joy to introduce our speaker for this session to all dignitaries and delegates. Professor Dr. Vandana Patravale, Madam. She is currently the Professor of Pharmaceutics at the Institute of Chemical Technology, Mumbai. She has around 30 years of prosperous teaching and research experience. Dr. Patravale has been active in teaching, research, and service throughout her career. She has over 200 referred publications with over 8,277 citations, H-index of 46, ITN of 111, 17 granted patents, 22 patents in pipeline, and 2 trademark registries. She has published 2 books and 25 book chapters with international publishers. Her areas of research include development of nanocarriers with major emphasis on infectious diseases, cancer, and neurodegenerative disorders, medical device development, nanodiagnostics, and nanovaccines. She has been bestowed with various honors and accolades. She was awarded Fellowship of Indian Chemical Society, Kukreja Oration Award, APTI's Dr. Manjushri Pal Best Pharmaceutical Scientist Award, Sri Amrit Modi Distinguished Researcher Award, Bill Melinda Gates Grant Award, Best Pharmaceutical Scientist Award, Veneto Nanotech Award, APTI's Best Teacher Award, Fellowship of Maharashtra Academy of Sciences, and K.H. Garda Distinguished Researcher Award, to name a few. She is Vice President, CRS Indian Chapter, Editorial Board Member, DDTR, Editor, CRS, and APTI Women's Forum newsletters. She is actively collaborating with researchers as well as industries within country and abroad. Has completed Indo-Swiss, Indo-Japan, and Indo-UK projects. She is executing all major grants from Indian government focusing on the product development. She has transferred about 20 technologies to industry, including drug-eluting stents being marketed in more than 65 countries. She is a true idol for all academicians and researchers in the pharma fraternity and a true hero to all her students globally. So may I now request ma'am for a presentation on nanotechnology in diagnosis, prophylaxis, and treatment of infectious diseases. Please, ma'am.
Thank you, Shalaka Madam, for a kind introduction. And definitely at the outset, I would like to thank Principal John D'Souza, sir, for giving me this opportunity, along with the coordinators and organizing team. A wonderful theme selection, I would say, need of the hour. So let me share my screen, and I would also like to seek blessings from Tatyasai Koreji. It is his death anniversary today, and elders always, we need to respect them and take blessings from them to prosper. Is my slide visible?
Yes, ma'am, it's visible.
Okay, thank you so much. Yeah, so as said, the title of my today's talk is Nanotechnology in Diagnosis, Prophylaxis, and Treatment of Infectious Diseases. So what I'm going to do is take some of the case studies wherein we have worked at ICT, and all of them belong to infectious diseases, and that's a lesson which needs to be taken. Though the theme focuses on viruses, all these technologies which we have developed are platform technologies and can be used for any of the infectious diseases, be them from bacterial source or fungi or virus for that matter. Am
Mr.
I
Rohit,
audible?
Mr. Rohit Tarada.
Okay, so no presentation is complete without showing this premier institute, that Institute of Chemical Technology, of which we are a part. And we are really blessed to be in this particular institute, which has given 19 Padma awardees to this nation. An idea of showing this picture is that this is Mumbai campus, but now we do have two more campuses, one at Orissa, Bhubaneswar, and another one at Jalna, Aurangabad. So our research on infectious diseases is focusing on diagnosis, prophylaxis, and therapy. And as you know that all these areas are very, very important now. If you look at the Indian scenario, there are huge number of Indians who die annually due to infectious diseases. There are various reasons for it. It could be the dual burden, that stress on the underfunded and under-resourced health systems. It could be because diagnosis is not complete and infectious diseases go unchecked. And then they begin to show a resurgence. And in fact, talking with our doctor friends, it will be very surprising for you that though we said leprosy incidences have gone down, when you talk to them, you get to know, in fact, they are again on rise, but they are going unchecked. And you know how contagious these diseases can be. And now COVID-19 pandemic has put even more strain on our stretched healthcare resources. So this particular slide just depicts that you can get many diseases, infectious diseases, which could be mosquito-borne, airborne, waterborne. And in fact, if you look at Maharashtra, where we belong to, you will see that we have all kind of infectious diseases, including malaria, dengue, you have typhoid, you do have hepatitis, gastroenteritis, and conjunctivitis, which generally begins when the rainy season starts. So that means lot needs to be done still, though we feel that we are sitting in urban settings, but we, the population who are in urban areas, are much more prone, I would say, than rural areas for many of these diseases. There is rise of infectious diseases, and majorly it is the increasing urbanization and human mobility. In fact, if you see this particular slide, what I'm trying to show you is a Mumbai local train station and a metro. And this is researched, and it has been shown that nearly 40% of the population of New Delhi has been infected by dengue virus, or would be infected at least once in their lifetime, which is a big, big number. And if you talk about metro in Delhi, we are at least 3 times in Mumbai, the population wise. So obviously you can predict what it is, what would be affecting us. Look at this particular depiction, which shows global examples of emerging and re-emerging infectious diseases. So those which you can see in red are newly emerging. Those you see in blue are re-emerging. And you will be surprised that all kind of infectious diseases are on rise. In the re-emerging class, you do have cryptosporidiosis. You have all kind of E. coli infections, some of the pulmonary syndromes. You know of the Zika virus. And talk about all the viruses, Nipah or Hendra or influenza, and now resurgence from MERS COVID to SARS. So these are some of them which are listed, along with areas where they are mostly, affecting the populations. And you can see one black dot too, which indicates deliberately emerging. Let's not talk about the deliberating emerging ones. Bio wars, is something which we all are scared of. So keep in mind that there is lots which is happening. Some of the parasitic diseases also come under infectious diseases, including malaria. And still, every 35 seconds, one death happens in Africa. Our areas, Orissa, etc., are again getting affected. Cerebral malaria is on rise. That means P. falciparum infection is on rise. P. vivax is a general common one, but P. falciparum is more dangerous, which can lead to coma in certain patients, leading to death. It says that we have a lot of challenges which need to be tackled. And these challenges are due to emerging or re-emerging infectious diseases, which you have seen in the last slide. It is due to lack of medical countermeasures. If you talk to people who are in the rural area, especially the physicians, they will tell you that they have to spend money from their pocket if they need to treat patients. So though government is trying their best that doctors reach these areas, the medicines don't reach. So we need to work a lot at that level. There are social stigmas which are associated with many kind of dosage forms. People are not ready to take them. For example, suppositories, which could be a best system for pediatric use. But it is not so popular in India. In fact, in Europe, if the kid is unwell, has fever, first thing mother would do is insert a rectal suppository, paracetamol. But in India, no more. There could be reasons for that. And one is the storage stability of those suppositories. So can we not work in those areas and come up with bases which can be used, which are stable at room temperature? They do not need refrigeration, and still tackle. That will remove fear of needles from, everyone's mind. So if kids can take, obviously adults can also take. Human-animal interface, we need to understand this very well, because there are many infections which spread from animals to humans and vice versa. And one such case study I'll be taking of brucellosis, which primarily affects animals, but from animals through their dairy products, it affects human beings also. And those who are working in close contact with animals, especially the farmers, will get infected. Not only them, if human beings are admitted actually in hospital, it's so very contagious. Symptoms are like malaise, fatigue, which are normal symptoms for many, many diseases. So even doctors get affected unknowingly. We need to look at different geographic factors and how different areas have different kind of diseases, what can be done to take care of them, and many more, of course. So moving on to the first case study, which is brucellosis. And as I told you, we've been working in this particular disease under One Health concept. By One Health, I mean come up with a treatment modality which will be useful for animals as well as for human beings. And in fact, the projects which I'm going to give you as case studies are funded by DBT ICMR, the diagnosis part, and the treatment part by Bill and Melinda Gates Foundation. Reason being that we were aiming to give a total holistic strategy. One for diagnosis, another one for treatment, of prophylaxis also. So as I mentioned earlier, brucellosis, if you know, it actually affects milk-bearing animals. So if cows and buffaloes get it, they need to be slaughtered. And in fact, from these animals, through their dairy products, be it milk, cheese, yogurt, anything, it will get transmitted to human beings. It can enter into humans via any wound if they have on their skin. Could also be because these animals need to be vaccinated. So by mistake, if the needle sticks during vaccination, can also lead to this infection. So infectious agent here for brucellosis is Brucella abortus. Transmission, already I have told you, if you look at epidemiology, more than 5 lakh new human cases of brucellosis are reported worldwide each year. And this is little old 2020 data. And clinically, incubation period is about 2 to 4 weeks. But the symptoms will last for 6 months or longer. That means how many productive hours of that human being are gone. So what did we do? We made point of care diagnostic kit. If you look at brucellosis, there are certain colorimetric tests which are non-specific. So they'll give you false positive or false negative results. On the other hand, you do have ELISA and RT-PCR. I think these have become common names nowadays. They are very, very expensive. Our purpose was, and our purpose at ICT always is, to come up with something which is very simple, something which is scalable, and something which is cost effective. So we came up with point of care diagnostic kit, which unlike the marketed kits, does not require blood sample. Farmers themselves, by testing the milk of animals or urine of animals, can predict whether the animal has this particular disease or not. So that was our simple rationale, because we understood the gravity of problem. Most of the times, diagnosis in animals is delayed because people have to do culturing to know whether it is brucella or not. And as I said, there are techniques like Rose Bengal test, complement fixation, agglutination, which are simple, but they give non-specific results, give false positive results, and all of them need serological samples. PCR ELISA, very costly. And this particular disease has zoonotic potential. That means from animals to human beings, there is possibility of getting infection. So what did we do? We came up with a very simple lateral flow platform technology. I call it a platform because we have actually patented it, and patent has been granted. What have we done? We have made fluorescent silica nanosensors. And what you can see in the slide is a lateral flow kit, which will have obviously the base on which there is a sample pad. Sample pad is generally made up of glass fibers. You can also use cellulose like polymers. But very important thing is that you should look at what is the porosity of this particular sample pad. Because when we talk about lateral flow assay, that means through capillary action, sample will move further. This kit has a conjugate pad, which again is made up of glass fibers or cellulose and has about 2 to 100 micron size. And this conjugate pad would be coated with a detector complex, which in this case is antigenic fluorescent silica nanosensor. You do have a capture pad where you will be actually putting the sample, and that is nitrocellulose membrane. In fact, again, there is a lot of potential existing in literature, so that if you can come out with alternative to nitrocellulose membrane. Nitrocellulose, I'm sure, as you are from pharma background, you'll know it is an explosive material. So storing it on the manufacturing site requires flame-proof areas. So the capture pad is typically 2 to 20 micron nitrocellulose membrane on which you will have two bands, two immobilized bands. In this case, one will be anti-Brucella IgG, which is a test line. And on the control line, you'll have anti-immunoglobulin, that is anti-LPS IgG2. What you need to understand is for what particular disease you are developing the diagnostic kit, understand the antigen, understand the antibody. There are two types of kits, one on which antigen is coated, one on which antibodies are. So I've already given you the composition of immunochromatographic diagnostic test strip, which has a sample pad, the conjugate pad, has a capture pad. In the capture pad, you do have the test line and the control line. And you can see that I have elaborated to show it's just a schematic diagram. And such schematic diagrams is the need of the hour when you're actually submitting any paper, research manuscripts. So it depicts that the control line has anti-LPS IgGs, and you can see on the test line, you do have obviously anti-Brucella IgGs, which will come in contact with antigen. Antigen is totally on the conjugate pad. So what happens in this case when you are applying a sample, which can be, as I said, since we have developed point of care diagnostic kit, it could be saliva, it could be urine. It could also be blood, not that it will not give results for blood. It does give, in fact, just to test our, to get proof of concept, initially we start with blood and then we move on, because we need to actually come out with what would be the concentration of these antibodies. For that, we need to do certain trial and errors. And nowadays, of course, using DOE, you can do that. So what happens is when you are putting a sample, it will move further. So if your sample is positive, both the lines will glow. But if the sample is negative, it is only one line which will glow. Why? Because when you are applying a test sample as a sample band, there's bicapillary action, antibodies are there, you have antigenic fluorescent silica nanosensors, they further travel. And if the sample contains Brucella specific antibodies, because that means that particular animal has got infected or human being has got infected, it will have produced antibodies. So those antibodies will interact specifically with antigen and this line will glow. Obviously, if there are no antibodies, this line is not going to glow. But in any case, a control line will glow because control line is the one which is containing anti-LPS IgGs. So in simple words, very, very simple technique. You can assemble all the components at your end. And in fact, what we have done is now we have used, this is just example of Brucella. But we having understood that can work for any of the infectious diseases, we have done it for dengue, we've done it for malaria. You can run these in parallel. So 6 such plates can run in parallel. At a time, you can know results of 6 diseases. In fact, whether, for kids, you have to do test many times, even mother, to know whether genetically anything is being carried or some viruses like cytomegaloviruses viruses are getting carried. So in those cases, it can be run a very, very simple, very, very cost-effective way. Looks little complicated, but it is actually very simple. Once you understand, you have a plastic case. On a plastic case, all you have is a nitrocellulose membrane. A nitrocellulose membrane, you have a conjugate pad, which is made up of polymer, cellulosic polymer or glass fibers. And all you have to do is give little attention to the test and control line. That means you work on what would be the amount of these anti-LPS IgGs and anti-brucella IgGs which you are going to put, so that it can detect even when the infection just starts. So yes, granted Indian patent on this. How did we evaluate? Obviously, before we actually went on to evaluate the samples from humans and animals, we had to do this testing on SDS page, wherein you are confirming whether the LPS stands for lipopolysaccharide. And lipopolysaccharide is an antigen used in this case. So lipopolysaccharide, if it is reacting with my fluorescent silica nanosensors, you will see that there are no bands. So in the panel 1 and 3, you do not see any band, whereas in 2, 4, 5, you do see bands, right? So M indicates a membrane protein. 1 and 3, which deal actually with fluorescent loaded silica nanosensor, as well as rhodamin loaded silica nanosensors. We made 2 types. So if someone is not happy with the green, we can give them red. It's as simple as that, meaning you can actually load any dye onto these silica nanosensors. Of course, there's a process for that too. And if you read our paper, we've already published, everything will be clear. So what did we do? We tested this ICDT, which is ICT developed diagnostic test, also stands for immunochromatographic diagnostic test, for sensitivity, specificity, and whether it is in agreement with the other results, say of ELISA, PCR, etc. So you can see that when we compared it for blood samples, it is 100% in agreement. And it is as sensitive as ELISA, as sensitive as PCR. And when we have tested it for urine samples also, it is in the same boat. That means we were able to test these human beings, and the ones, all the positive cases came from Nair Hospital here in Mumbai itself. And we could say that ICDT concurred with ELISA and PCR results. Majorly, we had done this for animals. So, lot of input data, 103 cattle, 30 buffaloes tested. Note to the farmers, it is really a loss if they have to slaughter their own animal, and that also milk-bearing animals. So you can, relate with the farmers. In fact, now this particular testing is going on in Netherlands and New Zealand both. Because the results are so very good, commercial partner has already taken this kit from us. So you can see that ICDT, even for animals, you look at the serum data or saliva or urine, at all levels, it is comparing well with ELISA and PCR. And the other techniques, which are agglutination technique, as I told you, or Rose-Mangal test, they are not that specific, though they are cheap. You can see that it is not definitely very sensitive and specific. So we could test these animals. First, we tested from blood, then milk, then saliva, then urine. In all cases, we could show that it's very, very easy to take this particular kit ahead. So novelty of the method, yes, definitely our silica nanosensors is a novelty. Non-invasive method we are using, what else is required? Farmers themselves can test. So as compared to $400 to $500 which are required on kit. You can see the cost. I've already put this competitive landscape, because such presentations we need to give when we are presenting our data to industrial partners. So we show the technical advantage, but we also show cost advantage. So cost advantage, definitely you can see over ELISA, which says from $420 to $527 $715. You know what is the raw material cost for our kit? ₹5. How much the kit will cost? 20 rupees. Obviously, any farmer can really make use of it. Obviously, now since it's gone to industrial partner, all industries are business minded. So even if they give at 100 rupees, it's still worth it. So key differentiators, you can see that I have compared all different techniques which are available, but major advantage of our kit is that can really use non-serological media, milk, saliva, and urine, and definitely can eliminate blood withdrawal totally. Taking this case study further, as I told you, this particular platform of fluorescent silica nanosensor lateral flow assay can be used for any of the infectious diseases. So we have got grant from DBT BIRAC for COVID. But again, for that, we need to understand, we wanted to make it still simpler, in fact, and we have proposed a colorimetry test, not even fluorescent. Fluorescent technique, at times may need at least a UV lamp, but colorimetry will not need even that. And the results for that are also positive. Of course, the test results are not presented here because we are still in the optimization stage. Fortunately, when we are using, I would say, the reagents which are procured from Sigma, we are getting positive results. Unfortunately, when we take the same reagents from our Indian counterparts, we are not getting the required color intensity. We do get a color change, but not the color intensity, which, very rapid test. We are proposing it as a 5-minute test. So still working on it. So maybe next time we'll be able to present this data. Okay, the next one is the prophylaxis part, wherein we have developed intranasal nanovaccine. Again, when we talk about vaccines, you know that cold chain transport is one major drawback, because of which the cost of vaccine also goes high. That's the very reason why Pfizer vaccine was not very popular in India. In fact, initially it didn't get permission, because finally, in our municipal hospitals, maintaining that -20 or -40 degrees Celsius would have been a real difficult thing and a challenge, leave apart the rural settings. So idea here is that we will develop vaccine, which is a nano vaccine, which would be given by intranasal route. And we'll make this using a green technology, which in this case, what we are using is supercritical technique. And I'll show you a schematic to explain it. Just before that, I've already told you, human beings have the symptoms of malaise and fatigue for about 6 months. They feel no energy at all. And what is the treatment given to them? They are unnecessarily loaded with antibiotics and they do no good. They further produce weakness in human beings. And can you imagine rifampicin given as a monotherapy even to pregnant women if they come in contact with this disease? What effect it will have on kids? So right now you can say that there's hardly anything which is available. For cattle, they have to be slaughtered. Already told you, they have to be isolated, culled, or slaughtered. This can affect even sheep, it can affect yak, it can affect any milk-bearing animal. So that means once you have the diagnosis, and if you have a prophylaxis, that means if you have a vaccine, which again farmers themselves can administer, it would be a boon. Because right now what exists is live attenuated vaccine. Again, that live attenuated vaccine storage condition is refrigeration. And when people go, to give first dose, by the time they go to all the villages, it's time to give the second booster, first booster, so on and so forth. So it becomes really difficult to control the population which is getting infected. So we thought here lies a challenge. And can we look at making something which is very simple? So believe me, all these something very simple comes from our lab students. So I will not take credit for that. We do brainstorm. So everyone is told to read about what the disease is, what currently exists, and what can we make which can actually basically bridge the gap and be very simple to scale up. So we thought, let's not give anything IV, because again, there'll be some same problems. Animals, if you have to give injection, it is very, very difficult. And those of you who have seen, I personally went and saw how this vaccine is given, just to understand, it's really very, very difficult to handle large animals. So in those cases, nasal would be a boon. So our objective was to develop subunit vaccine. Subunit vaccine meaning you are using some particular fraction, right? Here we are using LPS, which is antigen. It would be made up of submicron sized particles. Submicron meaning they are nano. Mucoadhesive, because you are giving it by nasal route, we know that nasal mucosa has antigen presenting cells, which will take up, but we want the action also to last for longer period. An idea is, again, the nasal secretions are there. So it shouldn't happen that it just gets washed away. So prepare subunit vaccine, which is submicron size, mucoadhesive, and biodegradable, to be given by nasal route. So rationale was deliver antigen in the nano form using nasal route, but making it using a platform which is eliminating the total need for use of hazardous organic solvents. One step, and that's again why we got this patent granted. So what have we done? I've identified again here something which is from the indigenous source, it's from tamarind seed. So xyloglucan is taken in a chamber and you're passing carbon dioxide. Now this carbon dioxide is pumped at conditions which makes it liquid. So it's a gas, as you know, carbon dioxide. So at supercritical conditions, it makes it liquid. So it's pumped at pressures. Temperature is less, so nothing happens to the antigens. And then when carbon dioxide solubilizes this particular polymer, that polymer again could be with your antigen, it could be with your immunoadjuvant also. Allow it to pass through a small nozzle. So when this is pressurized coming out of a nozzle, what you'll get would be nanoparticles. Either you can collect solid nanoparticles. In our case, we wanted to make a nasal vaccine. So we've collected it in a vehicle so that it's ready to use formulation. If we were collecting these particles, again resuspend them, again stabilize them, would have been a big issue. So simple, one step, manufacturing using REST technology, which is nothing but rapid expansion of supercritical solvent. So what have we done? Expansion of carbon dioxide, which solubilizes these things through this orifice. So this just shows the patent which has been granted. We have characterized it adequately. So SCM as well as DLS, always we do both just to make sure, that many times dynamic light scattering results can be misleading. So you need to confirm it. So using SCM, we confirm that it was submicron. Submicron meaning less than 1 micron. 1 micron is 1000 nanometers. So you can see that though you are getting biphasic pattern, they are all under 1 micron. We also tested it for mucoadhesion. And NV50 is our vaccine candidate. You can see that it has given work of adhesion which is much more than vehicle alone. And that is why, because we have selected a polymer which not only encapsulates your antigen, but is mucoadhesive too. In fact, we have many patents around this particular polymer for other diseases also. Once we made these particles, we tested stability studies. Now we do have even 36-month data, and the results with respect to particle size remain the same. LPS content is antigen content, and you can see it is very important. Many times people forget, just like you have drug nanoparticles, you are doing drug content, here you have to do LPS content. And if you added immunoadjuvant, because I was focusing on indigenous, simple, cheap raw materials, I have taken pulaya-saponin as an immunoadjuvant. And you can see that particular immunoadjuvant also needs to be quantified. Once we made this particular vaccine, we started with in vivo immunogenicity study. And obviously, in that case, you are looking at antibody titers. And you can see that serum IgG, IgG2A, IgG1, all the levels increased with our vaccine candidate. Not only that, we also looked at alveolar fluid. Why? Because you are giving this by nasal route. And if you are saying that nasal mucosa takes it up from nasal, it will enter into respiratory tract. So whether it is giving prolonged immune response, there also had to be seen. And we were very happy to get this response. And can you imagine? This is the result which has again given us a very big, huge grant from DPT BIRAC for COVID. Because we are showing that we have a platform which can be given by nasal route, can definitely rule out cold chain transport and give us such good antibody titers. Having done this, next step was to look at in vivo protection study. Obviously, such a study has to be done in class 3 setups. And I've been requesting Professor John D'Souza for setting up class 3 facilities in TechECP Varnanagar campus. Believe me, this study was sponsored by BMG, Bill Melinda Gates Foundation, but 1/3 of the money went back to USA because in India, lead time to do our study was 1.5 years. And this full project was only for 1.5 years. So I could not wait for that long. So I had to do it in collaboration with Weiss University, USA. And this is the protocol where you are administering by nasal route nanovaccine. 28 days, you are giving 1 booster. 56, you are giving a challenge. That means by B. abortus, which is a causative organism for brucellosis. And then at the end of 84 days, you are looking at anti-LPS antibodies. And of course, you are looking at even the histopath. Whether the vaccine platform is safe or not needs to be seen. And also, this can form reservoirs in lungs, liver, spleen. So you can see that this is absolutely same like COVID, which also goes and affects lungs majorly. So that's the reason, again, why the grant came through very easily. So this is how we decided what we will administer, on which day, by which route, what doses, and started with our study. Before we started with our study, the professor there wanted to check cytotoxicity studies. So we said, go ahead. Just to make sure, because he had not heard of TSP at all. So he was little hesitant that whether such an indigenous molecule, it shouldn't happen that normal cell it affects. So obviously, when we did MTT assay on Vero cells and looked at its viability, you can see that it's 100%. You take any component, doesn't adversely affect. So we went ahead and then looked at what were the results. And this slide very clearly indicates that we have taken groups, which is only buffer. You have immunoadjuvant with TSP, which is xyloglucan, placebo. You have only antigen. You have antigen and your immunoadjuvant, and of course our nano vaccine. So in case of our nano vaccine, significant increase in IgG1 as well as IgG2A titers was seen. And in fact, not only that, it shows a very gradual rise. And in fact, that gradual rise is what is important for memory effect. So this gave us confidence that we can move ahead and look at what happens even in bronchoalveolar lavage samples. And very clearly you can see that in bronchoalveolar lavage samples, LPS has not given any IgG1, but our nano vaccine candidate has definitely given us good titers. Statistically significant production of IgA as well as IgG1. So we moved ahead, looked at whether colony forming units reduce, because that is the purpose. If infection has spread, it has induced colonies to get formed. So CFU reduction per gram of tissue, you can see in all cases, nano vaccine group is giving us the maximum reduction, CFU reduction per gram. So this is given us a proof of concept that definitely bacterial burden is going to reduce and no splenomegaly seen by giving our vaccine candidate. In fact, that professor was very happy and he's using it for other diseases too, and here we are using it for COVID now. So this case study has shown you that you can make a green vaccine. You can use simple ingredients. And in fact, when we presented this to COVID task force, they said it's okay, you can go ahead with this platform. But meanwhile, it would be good if you take some regulatory approved polymer and do the studies. So simultaneously, one project has actually got culminated into two. And now with newer and newer variants of COVID coming, it is going to culminate probably to 4 or 5 projects. So this is a project which we'll be doing now with NIRRH. So we have done this vaccine. We have done PLGA-based vaccine also for nasal use. So both are ready and animal studies are on. So we are waiting for the results. So one, diagnosis shown, second, prophylaxis shown, and third case studies for a parasitic disease, which is for malaria. Again, what I'm trying to tell you is keep your systems simple so that industry can take it. So obviously, when you look at number of malaria deaths which are happening, obviously it's Africa which is predominating, but India also has about 2% share. But when you talk about overall 3.2 billion people, which is half of world's population, is at risk across 47 countries. In fact, 2019 data, this is slightly old data, you can see that 409,000 people died from malaria. So obviously in Africa alone, there is loss in economic productivity. And as I've shown you in the initial slides, metro cities are at risk. Now what happens when malaria affects you is your normal RBCs, they become granular. Why? Because a malaria parasite has invaded. I'm sure all of you are aware of the liver stages and the blood stages of malaria. So malaria parasite multiply inside RBCs. And as a result of which, if it reaches on the brain side, all your blood vessels become hardened. And through those hardened blood vessels, the blood cannot flow. The net result of cerebral malaria is patient becomes comatose. So if they are not treated in time, they will die. And if you want proof for that, read WHO guidelines. WHO guidelines give you what are the modalities for treating malaria. Artemether lumefantrine is a combination which is used. It is orally given, but for oral absorption to take place and give you effect, by then the patient might just die. The next good available thing is artesunate injection, which you'll see in my further slide is no good. So again, when you read these WHO guidelines, you'll note that mortality from severe malaria, which is cerebral malaria, approaches 100%. Now this is the real unmet need. So mad as we are, we said we'll take up this challenge and see whether artemether lumefantrine can be given as IV dosage form. Because if it is given by IV route, obviously patient can be saved. What were the challenges? Both the drugs are insoluble. Lumefantrine is very high dose drug and insoluble. That was a challenge, but we said, can we, by reducing it or nano-sizing it, make a nano drug delivery system? And we succeeded at that. I'm just showing the results. What we have done is we have prepared nano-structured lipid carriers, where now we have mastery, about 2.5 decades of work in that direction. And when we did these studies, you can very clearly see that parasites do not rise at all when you are giving, say, monotherapy, which we published long back. 2014 paper, if you read, you will get even the mechanism, why is this happening? So when we gave the combination, this is with monotherapy, it is taken care, but monotherapy, we started with monotherapy, but WHO has banned monotherapy now. So if you have to give 2 drugs in combination, what 2 drugs can you give? We said, why not start with ARM and lumefantrine? And you can see that it gives sustained release of the drug. Again, if you are developing any kind of anti-malarial combination, see that either one drug is short half-life, one is harder, longer half-life, or one drug acts by one mechanism and the other acts by another mechanism. That way you can look at whether they are getting synergistic effect or not. So we developed NLCs which had size less than 100 nanometer. And when we tested them in model, which is known as Peter's 4-day test, wherein you are actually giving P. burgei. P. burgei is the one strain which affects animals. P. vivax doesn't affect animals. So it gives you, P. burgei will give you symptoms just like what human beings will. So you give P. burgei and you also start the treatment simultaneously. You can see that RNLC at 1/5, 1/20th, 1/50th doses, all doses, it has shown complete clearance of parasitemia. That means 0% parasitemia. Whereas this is a marketed one, which goes up. But when we looked at the treatment module, no, just like in malaria, you have symptoms, fever, chills, and then you take the drug. Same way, we said, okay, let parasitemia develop and then give this particular combination by IV route. You can again see that our marketed artesunate has shown recrudescence. By recrudescence, you mean at the time for which you are giving treatment, parasitemia is under control. The moment you stop, parasites increase. So what is the use of such a combination? Ideally, if you are given treatment and you leave the animals, you can see in our case, at 1/10 of the therapeutic dose, all animals have shown complete clearance. So we went ahead with 1/10 of this dose and looked at cerebral malaria model now. And cerebral malaria, what happens? You have to use C57 mice. You cannot use biopsy. So use C57 mice, record the rectal temperature. 50% of those mice develop cerebral malaria. And again here you can see when we did the testing, marketed one is showing recrudescence, very high recrudescence. Whereas our treatment group has given absolute 0% parasitemia. And in fact, this again patent is granted, technology has been transferred, clinical studies are going on. There are some delays because of COVID. What I'm going to show you in these videos is what I just talked about. We talk about selective uptake of these NLCs. So these NLCs, which you can see in the picture, are red color. That means we have loaded them with rhodamine and then remove the free dye completely by dialysis. And when I start the video, you will see that these ones are the normal RBCs, and wherever you see a granular mass are the infected RBCs. Because RBCs on their own do not have nucleus. I hope everyone remembers anatomy. When they are infested by parasite, you'll see a granular mass. So when I start this video, you'll see that this is a live imaging done. Our rhodamine NLCs go only where you see a granular mass. So they are selectively being taken up by infected RBCs. And when we did the studies, I show you marketed group first. As I told you, the study has to be done in C57 mice. So this is after treatment. After treatment has been given, typical symptoms of cerebral malaria is, when I say patient gets comatose, that means animals will behave as if they are paralyzed. So you can see that these animals hardly are moving. If at all they are moving, their gait is affected, their health is affected, temperature rises, and this is after marketed formulation has been given to them. Whereas this video will depict how our treatment helps these animals. So in fact, if I have to believe the veterinary doctors, they said that they become so healthy that they could be used after a washout period for another study. So this shows that we have something for an unmet need. So is that unmet need only malaria? No. In fact, we have done n number of tests to show that this particular formulation will be effective to reach brain for any kind of such disease like sickle cell anemia. It could be, there are many motor diseases which affect patients. So we are now looking at potential of the same NLCs for other diseases, where what actually they have done, obviously they have ARM and LFN definitely is helping to clear parasites. But by doing certain biophysical tests, we have shown that they restore the flexibility of the blood vessel. So we've done those studies at TFR and shown that it restores the flexibility of blood vessels. So that is required in many disease conditions. So that means it can be used for many diseases. Again, you know that infectious diseases like HIV, they have their own reservoirs in brain. In fact, recently I was discussing with scientists from AICTE, in fact, again, an oncologist, and she said, along with cancer, because the patients are becoming immunocompromised, why don't you look at TB? Because TB and cancer is a very deadly combination. So when we say that we have something which reaches brain in higher amounts, we should look at a spectrum of diseases. Do we have a little more time so that I can show another presentation?
Yes, ma'am. Yes, ma'am. We do have time. You can share your presentation now.
Thank you. Just give me a second. The second presentation actually is on novel applications and how, in fact, future is shaping up and what we need to do actually to gear up. Can you confirm if it is seen?
Yes, ma'am. It's visible.
Oh, sorry. Visible now?
It was visible, but it's not visible now.
Can I start, Shalaka madam?
Ma'am, it is not visible.
Okay, thank you.
It isn't visible yet.
Okay. So here you can see that biotechnology market definitely is increasing. And if you look at the statistics, nearly in 7 years, it's projected to double up. So from $400 billion, we are reaching to about $800 billion. And within this also, of course, US, China, and India markets are the ones which are growing. As far as application landscape is concerned, biopharmacy segment shares about 61.7%. Bioindustrial sector is growing at CAGR of 9.3%. These are top 10 biotech trends and innovation, and here you can see that people are working in what areas? Be it tissue engineering, bioprinting, synthetic biology, of course, biomanufacturing and biosimilars you've been hearing. Now we are talking about precision medicine, gene editing, and so on and so forth. So I thought,
Ma'am, your screen is not visible.
Screen is not visible.
No, ma'am, it isn't visible.
I will stop presenting then. Now?
Yes, ma'am, it's visible.
Okay. So biotechnology in pharmaceutical sectors, if you see, key trend is you use nanoparticles in stem cell therapy. What is the market driver? Obviously, rising R&D expenditure. So we need to focus on nanotechnology research, and this market is definitely projected to grow at a very, very rapid rate. So as you know, nanotechnology is nothing but structural engineering of particles. And when we talk about biotechnology, mainly we are talking about genetic engineering. Nanobiotechnology is a sum total of these two. So a lot of efforts are going on in this direction for targeted delivery, for making nanobiosensors, so on and so forth. So this slide in a nutshell explains that you can use nanoparticles as cargoes. So obviously, you can use nanoparticles for encapsulation. It could be small molecules, could be larger molecules, like proteins, nucleic acids, reporter agents. Or if you want to use it for active targeting, you can tag different moieties. It could be aptamers, it could be antibodies, could be certain peptides, PEG, as you know, for making it stealth. You can also play with charge of these nanoparticles as well as their shapes for selective targeting. And nowadays, just like you had control release stimuli responsive systems, now you have nanoparticles also, which will release the cargo based on external stimuli. What's happening in this area? All this time, people were working in nanoparticles, ribosomes, related systems, lipid systems, but now people are working on nanovesicles. Why nanovesicles? Because they are very highly biocompatible. These are the vesicles which you get from your own blood sample. So we are moving slowly towards personalized medicine. Also, production yield is 200-fold higher than some of the nanoparticles. They will have all the advantages of earlier systems too. So for example, if you say that liposomes could have functional targeting moieties, these vesicles, which are also termed as nanoghosts or exosomes, will also have all those advantages. So if you look at market trends, you have companies like Gilead and Gritstone coming together. For example, they are partnering for HIV vaccine, wherein Gilead is going to use its antigens and Gritstone is going to use the vaccine platform, which amplifies mRNA. And of course, it is using the viral vector. So likewise, can we pharmacists not identify biotechnologies? Or in fact, pharmaceutical biotechnology is one subject which definitely needs more attention in today's era. And can we ourselves not formulate success? Can you visit this BioNTech platform later? I'm just going to take one case study, but this platform from Germany, you will see, has n number of platforms. And if you read, you would know that all of it we are capable of doing, we the pharmacists are capable of doing. So what is done in this case? Individual patient samples are taken, be it blood, be it tissues. They are mapped for mutations. And then there is a prediction of neoantigen. After doing that, you are tailoring the RNA and then using it for immunotherapy. So obviously, meaning if there is a cancer, what particular biomarker is overexpressed or which gene will get overexpressed is to be known so that you can devise such treatment. So there is one technology which is known as ribomaps. Concept is antibodies here are encoded by mRNA. So as an alternative to recombinant antibodies, this is being used. And they are using lipid-based formulations for these. The lead candidate is being used for multiple solid tumors. BNT141 is a code which has been given. Likewise, BioNTech has other platforms. They have treatment for different viral diseases. They have treatment for infectious diseases, treatment for cancer, many, many more. Another thing which is happening is Zolgensma by Novartis. This again is using adeno-associated virus vector-based gene therapy. Now, why am I showing you this? Our aim at ICT, I told you, is to simplify things. And we are moving away from these viral vectors. Our research is focused on using non-viral vectors. So can we use non-viral vectors and still treat diseases? So this is another snapshot from AstraZeneca and Ethris, who are working in area of course, respiratory diseases. Academic trend, I've already told you, people are generating SIRS from gram-negative bacteria, outer membrane vesicle, E. coli vesicle. You know that this is E. coli is responsible for cytokine release, which can lead to systemic inflammatory response syndrome. So on one side, this can definitely be extracted and you can use it for animal models. On the other side, what people have done is used as vesicles. Say human monocytes are taken. You remove the internal cargo, whatever is present there, and make membrane pellets. And these membrane pellets then can be loaded with what you want. In this case, I'm showing dexamethasone, but you can load even larger macromolecules. What are the results? Finally, you are getting drug-carrying nanovesicles. So what are you doing? By removing these nucleic acid and proteins, what you have is only the shell, which is a ghost. It's a ghost membrane. That's the name. Nanoghost also is given to these things. And these are known now to target endothelial cells very, very selectively. So confocal image tells you about that. So this particular slide is depicting how can you load ghosts with dexamethasone, and 10 pictures very clearly show that dexamethasone has got encapsulated here in these ghosts. And then when in vivo studies were done in mice, obviously when OMVs, outer membrane vesicles, were injected in these mice, it resulted in hypothermia and body weight loss, obviously resulting in inflammation syndrome. And now you can compare free dexamethasone versus ghosts which are loaded with dexamethasone. One could reduce the dose 4 times. So by reducing dose, getting the same efficacy, increasing the survival rate at the same time, these are less toxic. So can this not be used for pulmonary use in COVID? It can be used. Can it be used by IV route? Yes, it can be used by IV route also. Oral, of course, is still a challenge. So one case study, very quickly I'll take, wherein I'm discussing actually oncotherapy. Why? Because as I said, the doctor from AICTE said these patients are immunocompromised. So obviously, first you have to effectively treat them and then see whether you can load, co-load chemotherapeutic drug along with anti-infectives or chemotherapeutic drugs along with genes. So very quickly, I'll tell you one case study, which is for breast cancer, which is projected to affect 1 in 28 women in India. Statistics, when I see, it is really very, very disturbing. So what have we done? We have designed an active targeting system, wherein we have our own lipid systems, because I feel lipids are most compatible. Along with that, we have used a ligand. People have used all kind of antibodies, aptamers, many peptides, but what we have used is we have used Febodies. And as a linker, we have used PEG. So obviously, this slide is just telling you that when you are actively targeting, your nanoparticles will go and sit in the tumor rather than going all over blood and then going to all the organs, giving you side effects. Something about this Febody, these are engineered proteins. Advantage of using Febodies is that they are stable at high temperatures. They can sustain 90 degrees Celsius. Also, they can sustain variety of pHs. And that's the reason why we thought we'll use anti-HER2 Febody. Why anti-HER2? Because HER2 positive breast cancer is on rise. About 30% of the cases which are reported in cancer hospitals are HER2 positive. For them, the usual chemotherapy is not going to work. So then we synthesized the lipid, we looked at its toxicity, we looked at MTT-SA and found that we could use this particular basically lipid peg conjugate at less than 250 ppm and designed the system. So what have we done? Right now we put docetaxel, but again remember, this is just a model drug. But docetaxel, not that it is not used, paclitaxel, docetaxel are frontline chemotherapies. But what I'm trying to tell you is you can put any cargo here. It could be single drug, it could be single drug with anti-infective, it could be drug with gene, so on and so forth. So we have a linker and then we have Fe bodies. So when we looked at the stability in solution form, they were not stable, but when we freeze dried them, you can see that they were very stable. Next was to look at HER2 MCF7 cells, because breast cancer we were looking at, so MCF7, and specifically we were looking at HER2 positive cases, so it was important for us to look at these cell lines. And you can see that Fe nanos, which we call them, we've given the name Fe nanos, have given potency which is nearly 4-fold higher than the usual passive NLCs. And they are taken up within an hour by all the cells. So cellular uptake studies also indicated. These are on specific cell lines, please keep in mind. We have completed all the concentration, plasma concentration studies, biodistribution studies, looked at acute genotoxicity, mutagenicity, everything has been completed. So just waiting for, in fact, animal studies to begin. And the last one is gene delivery, wherein we have looked at prostate cancer. For prostate cancer, we have used siRNA, and all of you know siRNA are negatively charged. So that is a demerit of siRNA because it will be immediately thrown out of the body because kidney will filter it off. So what have we done? We have used nanotechnology again here. Again, if you look at the statistic, 60% higher risk for, again, Indians. See the estimated number of deaths. Now, every 2 minutes, a man is diagnosed with prostate cancer. Very, very alarming condition. Obviously, we know that prostate is a problem with the elderly people, but cancer, pray to God to keep away from that. So we talk about the gene delivery here. So we did a complete literature survey and found that we can look at c-MYC. Why MYC? Because it has direct role in control of DNA replication. And of course, upregulation of this MYC genes has also been found in other cancers. So we thought if we have a platform which behaves well, it can work for other cancers too. So in this case, we have formulated cationic liposomes of siRNA, and all you have to do is negatively charged siRNA. You need to use a positively charged lipid, which is used in a liposome to form a lipoplex, and then this lipoplex will be taken up by endocytosis by the cell membrane. So this is a lysine-based cationic lipid, again patented one. We have looked at all the characterization and then looked at hemolysis assay because it would be given, not being gene delivery, it has to be given by IV route. So we need to look at hemolytic potential. Obviously, simultaneously, we wanted to see whether this cationic lipid can be given by oral route. So we've completed oral toxicity studies also, and it is safe. So we have now something here which is safe to be given by oral route too. Then we have done cytotoxicity studies, and IC50 for this cationic lipid was 218 micromolar. That means you have to work at values which would definitely be effective. We characterize these liposomes. So when you have to characterize, obviously you can see you have to use a gel dock system. siRNA band which is seen should not be seen when it is got complex with your liposome. So you can see in panel 510 what is written, you cannot see any such band. That shows that you have to use these ratio to form a complex. We have completed stability studies. It is physically stable. We have looked at cytotoxicity studies. which are prostate cancer cell lines, DU-145. And conclusion was that at 10 nanomolar as well as 50 nanomolar, no cytotoxicity was observed. Then followed gene knockdown efficiency. Again, here we have collaborated with AICTE, and you can see that our system is able to be very, very effective. And it is the siRNA lipoplex, which we have prepared, is efficacious and has knocked down MYC gene in the cell line. Now, lipofectamine is a positive, the gold standard. Lipofectamine is also lipidic, but disadvantage of lipofectamine is that it cannot be given internally to human beings, very toxic. It has very good transfection. So what we have shown in these studies is we have a lipid which gives transfection as effective as lipofectamine without the side effects which are associated with lipofectamine. Likewise, we have variety of platforms. We've done protein blotting to show that lipoplexes definitely are giving you highest knockdown. And when another confirmatory test is, if they are knocking down, histone will increase. So you can see the siRNA lipoplexes have shown very high histone level. So here we conclude that we do have lipoplex as a platform again, which can be used for any kind of gene delivery. Of course, it can be used for drug delivery too. So with that, I would say our efforts are always, as our Chancellor of ICT puts it, we work in area of assured innovation framework. Affordable, scalable, sustainable, universal, rapid, excellent, and one which can be distinctive, unique. And another saying of him is that we need to get more from less for more, get more performance from less resources. People always say we do not have resources. Believe me, when I started, I did not have 10,000 rupee grant too. But you start so that you can reach to more people. So this is known as MLM technique, which is his funda of Gandhian engineering. So I thought I'll pay tribute to Tatya Saheb Koreji by giving some examples which are in line with Gandhian engineering. These are our major collaborators, and of course, without this family, I am no one. So I would like to thank each one of the members of VBP family for contributing to science and to identify unmet needs and work towards it. Thank you so much to the organizers for giving me this opportunity to share some of the work which we have done. I hope I am in time.
Yes, ma'am. Thank you so much, Dr. Vandana ma'am, for your enriching insights and guidance on the nanotechnology in diagnosis, prophylaxis, and treatment. You explained it with apt research case studies and made it very lucid for everyone to understand. It was very interesting session. Thank you, ma'am.
Thank you. Namaste, Mishra ji.
Good afternoon, Dr. Amit. You are muted.
Okay, fine. I'm unmuting myself. A brilliant talk, and I really congratulate Dr. Vandana for conveying so much in such a short time. And my congratulations and deep appreciation to her group, and thanks for joining us here.
Thank you. Thank you so much.
May I now request the participants for the question answer session. I request participants to please unmute yourself and ask your curious questions to our guest speaker. You can also post them in the chat box.
No questions?
I request the delegate
Yeah,
teachers.
Kiran sir is having question. Sir,
Yes.
you are not audible.
Sir, you're not audible.
There might be a problem in my.
Yeah.
Hello,
Now
is
you
that
are.
audible now?
Yes,
Yes,
it's
yes.
audible.
Madam, the talk is always wonderful. Some go out of my brain, but I try to assimilate it. As already I told you that some area regarding immunity and microbiology we should know. But still, there is just one question came into my mind that how, when we take milk sample for testing, then how sensitivity and specificity can be predicted. Maybe it might be give some challenges.
No, because it is finally antigen antibody reaction. You know, only how much volume of milk will be that has to be optimized. So, which in this case, Geeta madam, please mute yourself. Yeah. So, which has to be optimized, obviously, but more amount is always welcome.
Okay.
So there was absolutely no issue. In fact, I would say in milk there was no problem. For urine, we had to concentrate the sample. So that means the sensitivity from milk is much better than that from urine
Okay.
for brucellosis.
Actually, I thought that some of the immunoglobulins that will reach from the milk and that only used for quantitative estimation, and that orientation my question was there.
No, so
Okay, I got it. I got it, ma'am.
you are
Once.
using very, very specific antibodies, so nothing will interfere.
Okay, got it.
You don't have to
And
give
how?
milk, any kind of pre-treatment to milk. Nothing has to be done. Direct milk onto the sample pad.
Great. And how we can estimate or can conclude that it is a genetic transmission? How we can correlate with that?
Brucellosis, you are specifically talking about that, or
Yes, ma'am. With through this, I somewhere I heard this word. We can correlate the genetically transmission of the, or
so
maybe.
so far there is no literature which shows brucellosis is getting transmitted, from pregnant women to kids. But if it has to be tested, same kit could be utilized. There's absolutely no problem. Because typically what is being done in hospital setups is they are taking your blood sample, right? And then testing it, running through battery of tests. So CMV probably you were talking about. Cytomegalovirus virus is one which gets transmitted from mother to child.
Child.
So they are taking blood samples and doing the testing. So same test can be done. There's absolutely no problem. As I told you, now we are actually making a kit which will be able to detect 6 specific antibodies in one go.
Great. Always inspirational and something out of box work. Thank you very much, ma'am.
My pleasure. In fact, I think Rohit is currently working at TKCP, right?
Yes.
TKCP, sorry, has a huge share in my research, whatever results, data I'm presenting, I would say, because all the animal testing, we are doing it at TKCP. So obviously, kit part is not done there, but he is the one who's developed that methodology. It started with Dr. Swati Vyas, but finally, he's the one who's taken it up. So, in case you have some questions, you can always ask him also and interact one-to-one. Those of you who are at Vardhanagar, of course.
Ma'am, my question is very general. How difficult it is to work with biologics as in comparison with small molecules as far as nanotechnology is concerned? Means nanobiotechnology and nanotechnology with small molecules.
Yeah, so meaning I won't say, I always say nothing is impossible. So first thing. Second is, yes, you need to understand the biomolecule which you are working with, whether it is Some biomolecules are sensitive even to agitation. So whether it changes its structure just by agitation, if it changes structure just by agitation, you can obviously not put it in HPH. Again, so what we do is very systematically, if it is not reported in the literature, look at what would be the effect of external conditions like temperature, pressure, agitation, which are usually encountered in making nanosystems on the structure of that moiety. If there is no problem, like for example, Fe body, I told you, can withstand high temperature. So that's how we picked it up. That was the idea. So if you look at other antibodies, all of them are susceptible. So reading and reading, I think that's all what helps.
Yes.
Yeah, so challenge is only in understanding to what it is prone, if it is prone to degradation, pH, temperature, oxidation, agitation. Minimally you have to study and then start your work. So believe me, when I started working on macromolecules, I wanted to work on peptides and no one was giving me peptides. At that time, I didn't have enough grants to buy peptides. So what did I start? I started looking at, is there any drug moiety which is peptide-like? And that's how we started our work with the antibiotic, which is the peptide-like molecule, and learned a lot from that. So that helped us later. But yes, siRNA, if you are talking about working with these things, see that you do not work without wearing gloves, because even your enzymes present on the surface through your sweat also can degrade them easily. So those challenges are there. You need to be well versed with handling challenges. But other than that, entrapment efficiency is one thing which you need to work on. So
Yes, that was I supposed to talk on how much deviation we can expect.
So basically, yes, if small molecules you are able to load 70 to 80%. These moieties, if you have proper technique, you can also load 70 to 80%. I'll tell you that also. But you have to have a proper technique. So like, for example, one technology which we transferred from here to a Europe company is again called as Fe technology, wherein you need to understand, if we are talking about lipidic system, right? And you're trying to actually encapsulate hydrophilic moieties. So somehow you have to keep that hydrophilic moiety with the lipid core. So obviously, look at what is that component or moiety which has more affinity for lipid, though it doesn't give away your biomacromolecule, but keeps it with the lipid. So I cannot directly give you the technology, but definitely I think I've given you some hint, identify which are those components which can keep your moiety more in the lipid core if you're working with lipidic systems.
Maybe vesicles are very useful because of one of the reason is they're compatible with these all molecules too.
Yes, vesicle definitely. Only thing is, no, vesicle purification is a challenge. So that to a biotechnologist like you will not be a problem. I don't mind collaborating if you're thinking of something. But for us, purification right now is a challenge. Just to take Kiran's question, sorry, Kiran sir's question also a little ahead. Very soon to become doctor, so I cannot just say that. So what? Okay, milk ice exosomes also can be made. Can you imagine? Easiest vesicles from milk. Start working on it in case you have access to milk in abundance. So people have started already working on it, loading those. So such simple things should be done, I feel. So one thing is, if you do take it from milk and make vesicles, it would be useful for all those people who do not have lactose intolerance. Rather than if you're taking it from, say, for example, the case study which I showed, no, they've taken it from S. aureus and all, then it becomes very, very specific. So run your imagination wide and no one has touched delve into that area. That is my, I think, till the time I have good students, I'll go on doing that.
Thank you so much for the answer, ma'am. Thank you.
Thank you, ma'am. I would request the delegates if they have any curious questions. Any more questions?
I have tried to simplify my presentation, but in case it is not, you are free to ask me later too, no problem.
Yes, ma'am. We'll convey our delegates about that too.
Thank you.
Thank you so much, respected Professor Dr. Vandana ma'am. I believe this session would surely help all the budding researchers in contributing to quality research and publication in the field of infectious diseases and applications of nanotechnology in its development. You have surely paved the way to all academic researchers in this frontier by highlighting the Indian scenario in infectious disease with case studies and what challenges we actually need to tackle with the competitive landscape and market trends. It was surely a delightful presentation, ma'am, and you have evoked a novel concept in all the teacher delegates. On behalf of all of them, I assure that we will be looking forward in this new frontier. Thank you for your precious insights and sharing time with us despite your demanding commitments and busy schedule. Thank you, ma'am, for gracing the refresher with your presence. We would like to take this opportunity to felicitate our guest speaker with the appreciation certificate as a token of appreciation. So may I request Coordinator Dr. John D'Souza sir to please do the honors, and I request Professor Chavan sir for the display of certificate. Please, sir.
Thank you so much, ma'am.
Thank you. Thank you. It's always a pleasure.
Thank you, sir. Thank you, ma'am. May I now request all the delegate participants to kindly put your video mode on so we can take a photo with our speaker for this session, Professor Dr. Vandana ma'am. Please, delegates. And may I request Chavan sir for the photo session?
Yes,
Please,
ma'am.
sir.
Give some time so that people can settle.
Yes, sir.
Okay, sir. Done, ma'am.
Thank you, sir. Thank you all.
Keep
With this-
smiling.
Thank you so much, ma'am, once again. Shalaka, ma'am, I think we should start at 1 hour from now instead
sir.
of 2:30,
Yes, sir.
2:40.
Yes, sir.
Right?
Definitely. Yes, sir.
All
Thank-
should join at least 10-15 minutes before, please.
Yes, sir. I'll
Yeah.
announce that.
Yeah.
Thank you, dear all. Thank you, dignitaries and delegates. With this, we end with the first session. We will start the next session after lunch break, sharp at 2:40 p.m. Thank you all, and you are all requested to join 10 minutes before the actual time. Thank you, everyone. Have a great day ahead.